In this video, I'm going to be covering the key differences between meiosis and mitosis, and I'm going to be working through an overview of the process. And so the very first thing to understand when you're studying this stuff is the overall objective of both of these processes. With meiosis, our aim is to take some kind of diploid cell, such as a spermatogonia or an oogonia in males or females respectively, and generate 4 unique haploid gametes. And so let's dissect that statement a little bit. Haploid refers to the fact that we only have one chromosome copy of our alleles, and we're going to get 4 of them. And these 4 daughter cells are going to go on to mature into a sperm or an egg in females. Now, when we look at the process of mitosis, our aim is essentially completely different. Our goal here is to just simply replicate some kind of somatic, which is a body cell. So the cells that make up your skin or your eye that you need to regenerate over time because they just get old and they need to be renewed, they will undergo mitotic divisions. And so with that out of the way, the very first thing I'm going to start with is the first phase of meiosis, which is arguably where the most happens, and it is the most important phase in generating what is called genetic variation in biology, and that is called Prophase One. And we denote the meiotic phases with numbers because there are 2 phases. There's meiosis 1 and meiosis 2. Meiosis 1, we are dealing with a diploid cell, and what's going to happen here is typically if you looked at a cell in interphase, when it's not doing anything, its DNA is uncondensed and it's readily accessible by RNA polymerase so that you can transcribe proteins that you need to function. But when the cells need to undergo meiosis or mitosis, that DNA needs to condense, and it will condense into things called chromatin, which happens when DNA will wrap around histones that have lysine residues on them that are positively charged, and those positive lysine residues are going to attract and bind down the negative phosphate backbone of the DNA, and we're going to form chromatin, and that chromatin is going to coil and condense even more into visible structures that we can see with a normal light microscope under a slide, into structures that we call chromosomes. And in the case of these gamete cells that we're going to be generating, if we were looking at a spermatogonia, the father cell to those, what we're going to find is that these cells are going to have homologous pairs. And what it means to have a homologous pair means that you have 2 alleles for a particular gene. So if we were looking at a pea plant, for instance, what we would find is that this one cell is going to have an allele coding for a rough skin on the pea, and we might have another allele on the alternate homologous chromosome coding for the smooth skin on the pea plant. And so we're going to form these structures called chromosomes, and these chromosomes will have a pair that contains the same alleles. The alleles might have different DNA, but the alleles will code for the same gene or feature trait. And what's going to happen here is that these homologous chromosomes are going to overlap on top of each other, and they're going to form a structure called tetrads. And what is really cool about this is that they overlap so precisely that the gene loci are right on top of each other. And because the gene loci are right on top of each other, we form these things called chiasmata or chiasma, in which little chunks are going to break off from one homologous pair, and they're going to jump onto the other homologous pair, and the opposite process will also happen. And because of that, we are essentially generating genetic diversity during this process, and this is a critical process to life. This is how organisms and populations as a whole are able to spread beneficial traits throughout their populations. And this process is called synapsis. So this process during which we're breaking off these little chunks of the chromosomes and sharing them between the two. And the other thing that's going to happen here is that we're going to have these little structures called centrosomes. And if you're studying for an MCAT or another biology test, centrosomes are referred to as MTOCs, and that stands for microtubule organizing centers. And that's just a fancy way of saying that you're going to have microtubules emanating from these things, and these microtubules are made of alpha and beta subunits. And so that there is a plus and minus end. The minus end of the microtubules is going to be typically anchored onto these microtubule organizing centers, which in animal cells is just called a centrosome. And these centrosomes are going to migrate to opposite ends of the cell during prophase one. What is also going to happen here is that your nuclear envelope, which is made of one of the key structural elements of the cells called intermediate filaments, is going to begin to disintegrate. And so your nuclear envelope is no longer present, which means that your condensed DNA, your super condensed DNA now in the form of chromosomes, is going to be completely exposed in the cytosol. And this is going to be important for the next process. But the key thing to take home here is that a lot of stuff is going on during prophase one, and I would argue that it is arguably one of the most important phases of meiosis, and that's why we spend so much time studying it. And so what's going to happen next after we have our exposed homologous pairs and we've performed synapsis and whatever genetic variation has occurred, is we're going to enter the next phase, which is called Metaphase One. And also a keynote here, what's the difference between this and a prophase of mitosis? Prophase of mitosis does not have synapses. It has none of this going on. The only thing that's going on during mitosis, the version of prophase, is we're going to see the nuclear envelope disintegrating. We're going to have the mitotic spindles moving to different poles, and we're going to see the DNA condensing into chromosomes, but we do not have synapsis. That is an incredibly important thing to take home here. Now we're going to enter Metaphase 1. And in Metaphase 1, what is going to happen in meiosis is you're going to have the microtubules reach out, and this would be their plus end of the microtubules, and they're going to connect onto two things that are called kinetochores. And so kinetochores are parts of the DNA on the individual chromosomes. And remember, we have chromosome pairs, and so each chromosome within that pair is going to have its own kinetochore. So in this case, we're going to have two kinetochores on each of these. So that each one of these little dots right here represents a kinetochore. The microtubules will emanate out from the microtubule organizing center and then attach onto the kinetochores, and they're going to grab onto them. So you're going to have one coming from here, and you're going to have another one coming from the opposite pole of the cell during this Metaphase 1 process. We're also going to see the emergence of the equatorial plate, which is just a fancy name of the line that we see our homologous pairs lining up on. And what's going to happen next is, if you look closely here, you can see how some of these microtubules are not necessarily grabbing onto a kinetochore. What they are doing is they're connecting to each other, and then they're pushing off each other in order to help separate the cell like this. And so that is very important because we need to pull and push the DNA into the opposite poles of our cell during this process. Okay, and so a key thing that's going to be happening in the next phase of meiosis, which is called Anaphase 1, is that we're going to be ripping apart the homologous pairs, but, and this is the key distinction between meiosis and mitosis, these sister chromatids stay together. And so what that means is that these individual chromosomes are still in one piece, and they still have their one kinetochore on them, and that kinetochore is linked to the microtubule, and these are being pulled apart, but we've pulled apart the homologous pairs, but we have not pulled apart the actual chromosomes. And so by the end of Anaphase 1, what's going to happen next is that we're going to enter Telophase 1. And in Telophase 1, much like in mitosis, we're going to be reforming our nuclear envelope in both of our daughter cells now. We have two daughter cells. We're going to be replicating our centrioles for the next phase of meiosis. And we're going to perhaps begin to decondense our chromosomes back into normal DNA, so that if at this point in time our cells needed to perform any functions, they could, because the DNA is now accessible by RNA polymerase and the other enzymes required to activate gene translation. And so we also form a cleavage furrow here, which is just referring to an actin filament that is contracting down and pinching off the cytosol. So our one parent cell here is now two cells. And another important distinction is that after Anaphase 1, we are still left with a diploid daughter cell. We have two diploid daughter cells now, and that means that we have two copies of each allele in each one of these daughter cells. But our ultimate goal of meiosis from up here was to make haploid gamete cells, because these gametes are going to fuse together to form a diploid zygote that would have 50% of its DNA from mom and 50% of its DNA from dad. And so we're going to need to enter now the next main phase of meiosis, which is called Meiosis 1. And during Meiosis 1, we are going to enter, Meiosis 2. And so during Meiosis 2, we can see many more similarities between Meiosis 2 and mitosis. And so what's going to happen in this process is that we're going to have our prophase happen. And when you hear the words prophase, we should remember these terms. We're going to be having our nuclear envelopes disappearing, which is what we see. We're going to have our DNA that may have been decondensed during Telophase 1 now recondensing into chromosomes. We're going to have our centrioles migrating to opposite poles of the cell. We're going to have the microtubules beginning to form and emanate outwards. And the other key thing here is that each one of these daughter cells that was generated from Meiosis 1 is going to form two new cells. So by the end of meiosis, we're going to have 4 daughter cells, not 2 like you see in mitosis. Anyway, so moving forward with Prophase 2, which is very similar to what prophase of mitosis is, because in this case, we're not having any synapses occur as we did in Prophase 1. We can continue and move on to the next step, which would be Metaphase 2. And during Metaphase 2, we're then going to move on to our equatorial plate, and our kinetochores now are going to be connected onto the microtubules from the MTOCs. And during this process, what will happen is we're going to be ripping apart the sister chromatids. And that process is going to happen, the ripping apart is going to happen in Anaphase 2. Okay, and so one of the things that we've heard before with what some of the problems you can experience during meiosis is called Down syndrome. And so Down syndrome refers to a failure at this critical point during this process. And so what can happen sometimes is that we end up with things called trisomies. And so what trisomies refer to is at this point where we have, at Metaphase 2, we are still considered a diploid cell. And then after Anaphase 1, if we were to divide the cell in two, we now have two haploid cells. But if this process failed to adequately rip apart the sister chromatids, so let's say at this junction here, this microtubule broke, or the kinetochore failed attached on the right pole of this cell. What happened is the left cell here is going to be diploid for one of its chromosomes, and this cell on the on the right will be missing that chromosome. And so if at this process you had these daughter cells fused together, it would be a very big problem, because the fertilized zygote would have 3 copies of its genes. So it would have 3 alleles, 3 separate segments of DNA potentially coding for a protein, and this can cause Down syndrome. And so Down syndrome is referred to as trisomy 21. And so, assuming everything goes right and we do have a good separation of our sister chromatids, we move on to the final phase, which would be Telophase 2. And in Telophase 2, what we see is we have our microtubules in the microtubule organizing centers on the correct poles of the cell. We're going to have an actin cleavage furrow. So this actin is going to be contracting downwards and pinching off the cytoplasm of our 2 daughter cells. And we're going to be reforming our nuclear envelope. And a very nice way to remember telophase is it is essentially the reverse of prophase. And so we're going to be reforming our nuclear envelope. We're also going to be decondensing our chromosomes into chromatin, and finally into just normal accessible DNA stored as heterochromatin or euchromatin. And the euchromatin is loose enough that RNA polymerase can get to it and actually translate the genes or transcribe the genes and get the mRNA segments. And so this is going to wrap things up for the introduction and comparison between meiosis and mitosis. The key thing to note here is that by the end of this process, we have 4 unique haploid cells. And depending on if you're looking at a male or a female, this process does end up being a little bit different. In the process of the male, all 4 of these daughter cells can go on to become mature sperm and go fertilize an egg. But in the case of females, what we find is something different. We have things called discharged polar bodies, and what that refers to is the fact that all 4 of these cells that get generated within the uterus of the female will have the DNA content, but the cytosol volume will be significantly less in the polar bodies, and so except one of them. And so of the 4 daughter cells that get generated as a process of meiosis, 3 of them are going to have very little cytosol and they're going to be discarded, and then one of them will have a lot more cytosol and a lot more mitochondria and an endomembrane system, and that one will be the actual egg that can get fertilized by the male sperm to actually become a good fertilized zygote that can further develop in embryo development. And so this is going to wrap things up for this video. I hope you guys find it useful, and thanks for watching.